Up to this moment, 152.9 million people worldwide have already been infected with the COVID-19 virus, and 3 million people have died as a consequence of this fatal pandemic. The whole world has been affected directly by this pandemic, and its consequences grow each day that it passes without new solutions. The developed vaccines are bringing hope in favor to the end of this pandemic, but the rate of vaccination worldwide still is limited and not sufficient to end it. To consider herd immunity, according to experts, 80% of the people need to be vaccinated. Right now, worldwide, we only have 14% of the population vaccinated. These numbers highlight the importance of an urgent need for rapid and accurate diagnosis tests for COVID-19 detection and screening.
To solve this, we present you the COVID-on-a-chip phone immunosensor. This sensor can measure quantitatively the antigen levels of SARS-CoV-2 protein in blood in under 1 hour, as opposed to a PCR test which takes more than 4 hours and requires a lab, or to an antigen rapid test which only measures the protein qualitatively. The immunosensor is formed by a microfluidic chip and a sensitive smart potentiostat. The chip uses magnetic nanobeads dually labeled with TMB and detection antibodies of SARS-CoV-2 to measure the concentration of SARS protein. The procedure is simple. The patient places a blood finger prick sample and the labeled nanobeads in the inlet of the chip. Then they both pass to the vent and they are mixed in the waste reservoir pumps. After mixing in the presence of SARS protein in the sample, the SARS antigens bind to the detection antibodies in the nanobeads, forming a DMB immunogen complex. Then the complex reaches the reaction chamber. There, a magnet generates a magnetic field that attracts the nanobeads antigen complexes to the sensor surface, the electrode. On the electrode, the detection antibodies of the DMBs bind to the capture antibodies present in the electrode through the SARS protein. After this, the chip is placed in the sensor potentiostat, which generates a potential. Upon application of the potential, the nanobeads catalyze the reduction of the TMB. This creates an amperometric current proportional to the concentration of antigen present in the sample. The current's value is read in the phone and the concentration of SARS protein is calculated and displayed on the phone. If the sample does not have the antigen, the DMBs are washed away and no electrochemical signal is detected. Depending on the measured current, a different protein concentration is obtained. Our product has proven to be able to detect protein concentrations of as low as 100 picograms per milliliter in less than an hour. The best thing is that it can be performed anywhere without the need of a laboratory.
Our device is thought to be directed and used by a global public. The fact that it does not need the presence of experts for its correct use and diagnosis favorizes the use of it. Our amazing device can be considered to be used in schools, by the professors, in companies, by their workers, or even within families. Performing these tests continuously will facilitate a rapid detection of the COVID-19 virus, avoiding an increase in the number of new infected populations. We have to consider our actual competitors in terms of detection products, which mainly are PCR and rapid tests. We beat them in matters of time, efficiency, and accessibility. We also have to consider the new vaccines that have been created and that are being applied globally. As I mentioned previously, the rate of vaccination is still low, and moreover, according to the EMA, having received the vaccine does not imply that you can't spread the virus to other people. So, even when a high level of people vaccinated have been achieved, our product will be useful and necessary to avoid contact with those infected with the COVID-19.
In case you're interested in investing on it, let me tell you that selling the COVID-on-a-chip phone immunosensor will be an easy-peasy task. Right now, every government in the world is buying vaccines from pharmaceutical companies, and the same happens with test kits, which are needed in every country. To start the marketing campaign, we will get in touch with the Spanish government, which will provide the sensor to hospitals and pharmacies. In this way, patients will have their accurate COVID results fast, without having to wait for more than 24 hours. The second target will be big companies such as El Corte Inglés, MediaMarkt, and Carrefour to sell the device as an appliance for the general public. The last target will be online stores such as Amazon and eBay. Pharmacy and laboratories, GSK, Abbott, Etxebarne, Tinfa, will be in charge of synthesizing the nanobits and chips that will be sold separately, so that everyone can reuse their phone sensors. Thanks to the COVID-on-a-chip phone immunosensor, every person will be able to carry out their COVID test in an easy, cheap, and fast way from their own home.
Hello, we are Pablo de Anta and Eduardo Puchol, and today we will show you how the technology behind the 2019 paper Detecting Unamplified Genes via CRISPR-Cas9 can change the world. But before anything, let's quickly review some previous concepts. CRISPR-Cas9 is a recent technology that is starting to be used for diagnosis of genomic diseases. Based on the Cas9 protein and a strand of guiding RNA, we're able to search for the complementary strand of the guiding RNA across the genome and perform a cut. This then allows researchers and therapists to modify the cell's genome however they want with base pair precision. This is revolutionary, and since its discovery, we've been trying to make gene sensing and editing more accessible and easier than ever. But it usually has come with a few caveats, like the need for big specialized infrastructure, high costs, or the need to previously amplify the genes if we're looking to detect a specific sequence in order to get a readable signal.
A field effect transistor is an electrical component which uses a nearby electric field and its associated voltage differential to modulate the current flow, being composed by 3 terminals or electrodes, with a semiconductor between the source and the drain. Graphene field effect transistors take the typical FET device and insert a graphene channel tens of microns in size between the source and the drain. Being graphene a lattice of carbon atoms that is only 1 atom thick, it allows the FET device to have an extreme sensitivity, which can be exploited on a wide variety of applications such as photosensing, magnetic sensing, or biosensing, among others. While the carbon in graphene typically does not react or bind with most materials, bioreceptors such as amino acids, antibodies, or enzymes can be added through adsorption, or a linker molecule attached to a channel surface and allow other molecules to interact and impart an electronic transfer through the device.
Most methods for the detection of nucleic acids require many reagents and expensive and bulky instrumentation. Our device is a GFET sensor that uses CRISPR technology to digitally detect target sequence with intact genomic material.
Termed as CRISPR chip, the biosensor uses a gene target capacity of catalytically deactivated CRISPR-associated protein 9 or Cas9 complex with a specific single guide RNA or sgRNA, knowing this whole structure as DRMP. And it is immobilized on the transistor to yield a label-free nucleic acid testing device whose output signal can be measured with a simple handheld reader. The DRMP complex interacts with its target sequence by scanning the whole genomic sample, unzipping the double helix and associating upstream of a protospacer adjacent motif until it finds and binds to the target sequence that is complementary to the sgRNA molecule within the DRMP. The selective hybridization of the target DNA to the DRMP complex modulates the electrical characteristics of the GFET and results in an electrical signal output. CRISPR chip expands the applications of CRISPR-Cas9 technology to the on-chip electrical detection of nucleic acids. When talking about target-specific nucleic acid detection tools, most of the established methods for nucleic acid-based molecular diagnostic tests, such as PCR, have been optimized to amplify and detect target genome sequences. The majority of these nucleic acid detection tools are time-consuming and costly to use, as they require multi-step reactions, many reagents, trained personnel, and complex instrumentation. In addition, quantitative analysis of these nucleic acid amplification techniques often requires sophisticated probe and primer design and optimization methodologies for efficient targeting. Furthermore, they often still rely on large and expensive optical components for detection.
So the main pros that our product brings are the following. First of all, it is a low-cost and fully integrated compact nucleic acid-based diagnostic tool. Besides, this biosensor combines the gene targeting capacity of CRISPR-Cas9 with sensitive detection of GFET to afford a facile, rapid, and selective detection of a target sequence contained within an intact genome DNA. Finally, the immobilized DRMP is not only sequence-specific, but also it is programmable.
The main reason why we believe our paper can have such a great impact is because of the reach it can have. We are talking about a new way to check, quickly and cheaply on the field, any kind of genetic profile of our interest, from pandemic testing to checking for the presence of rare diseases. If we were to implement this method to detect sequences in the population, we could do testing on a massive scale and with massive depth. Most actual rapid testing methods either take a bit too much, are not precise enough, or function with principles that limit them, like proteins or antibodies. Up until now, we've only been able to check for the results of the diseases we were after. But with gene detection, it means we can pinpoint with the highest accuracy possible the sources of the diseases that we can find almost anywhere. Help us help everyone with two of the biggest technologies of the 21st century, CRISPR-Cas9 and graphene field effect transistors. Thank you very much.
Protein Corona, a new approach for nanomedicine design. We refer as protein corona to the protein adsorption complex formed on the surface of the nanoparticle. This adsorption modifies the diverse physiochemical properties of nanoparticles: size, functionality, surface charge, surface composition.
Protein corona is complex and unique to each nanomaterial nanoparticle. Furthermore, the composition and relative quantity of absorbed proteins may also not correspond with protein components and the relative concentrations in the exposure media. So in other words, we know that the protein corona is regulated by the protein and nanoparticle interaction and the protein-protein interaction. We can distinguish between hard corona, which contains the high affinity proteins, meaning they cannot detach, against the soft corona, formed by lower affinity proteins and is reversibly bound to the nanoparticles, so they can be detached. Therefore, small modification in protein structure caused by mutual interactions with the nanoparticles may induce an enormous impact in the pharmacological activities associated with the other proteins. The correlation between the nanoparticle size and the protein size is an important factor for determining the protein structures. So, the bigger the nanoparticles, the bigger the proteins will be attached. And obviously, the bigger the nanoparticles, the more the proteins will have to stretch to adapt to the shape. Common techniques to evaluate the nanoparticle size after protein absorption include dynamic light scattering, transmission electron microscopy, and sometimes atomic force microscopy. We have to highlight the fact that protein identification and quantity determination is best obtained through mass spectrophotometry. So, how do the exposure conditions affect the protein corona? We know that in the case of serum proteins, they reduce the interaction with the cells. For a higher time, we know that the rate of absorption obviously increments. For the fluidic conditions, meaning the pressure and the speed of the blood or the exposure, the protein variability changes, obviously. With temperature, the more temperature in biological ranges, the rate of absorption also increases. And what about the protein concentration? We know that the more protein concentration there is, the low affinity in large quantities proteins will take place for the high affinity in small quantities proteins. In the same way that the body fluids affects the properties of the protein corona, we know the nanoparticle properties also affects the protein corona. In the case of the factor of the size, we know that the bigger the size, the more proteins will be attached, but also the more deformation of the proteins, therefore the more instability in the protein corona. Smaller size will mean more stability of the protein corona. In the case of the shape, we know that the more surface there is, the more attaching points for the proteins. So we know the nanorods have a higher stability of the protein corona than the nanospheres. In the case of the charge, we know the charge affects composition of the foreign protein corona, and the more highly charged the surface is, the increases of the protein conformational changes. So we know that the higher the charge, the more cell intake there's gonna be. In the case of the hydrophobicity, we know that the higher hydrophobic the nanoparticle is, the increased absorbance of protein quantity and quality of charge obtained during the protein absorption. It increases the affinities of biomolecules and increases the protein conformational changes.
So, with all those information and data, a physiological prediction can be made. It means that what will be the biological and cell behavior outcome. Statistical studies showed that there is no relation between physiochemical properties of nanoparticles and the cellular response. However, the biological outcome is defined by the fingerprint of the protein corona. Moreover, the more adverse factors we evaluate, the more accuracy we will get in our prediction model. It is not the same to evaluate the protein corona fingerprint with 3 factors than do it with 9 factors. In this scheme, the prediction procedure is explained. The first step in every research with nanoparticles is the characterization, to know what we are working with. After that, we must evaluate the nanoparticle after the contact with a biological fluid, controlling what are the specific proteins that have been adhered. It means to determine the protein corona fingerprint. Finally, the nanoparticle protein complex is studied in terms of the interaction with cells, and the outcome achieved is added to the collected data. With a huge amount of information, a prediction can perfectly be done. So, with this prediction model, a specific modification based on the biological fluid and on the application could be more accurate. So, imagine that you have a car. a wonderful car, and you want to go from point A to point B. However, there is a snow condition through the trip. What will you do? If you take your car and you don't mind a snowy condition, probably your car will be full of snow in your wheels, and your car will destabilize and you probably end in the hospital. However, if you take care of your life and put snow chains in your wheels, you will avoid any problem with the snow and you will arrive to your desired point B. In this case, you are predicting what is the best solution to arrive to the point that you want. The car represents the nanoparticle. Now represent the biological fluid that will do some protein, some specific protein to adhere to your nanoparticle. And that we can create a library where we can search for any behavior of any nanoparticle for any biological fluid. I mean a library where you can press Ctrl F to search for the information that you want.
Thank you very much for your time, and we welcome any type of questions you may have.